Hello my dear students, I hope all of you are doing very well. My name is Abhijit. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. Today we actually continue our learning in Pharmaceutical Biotechnology Unit 2, where we are actually learning about the genetic engineering, rDNA, that is recombinant DNA technology or cloning, and how today I am going to tell you how you can use the genetic engineering in medicine. Okay. So, today I am going to tell you all the various aspects. I am going to tell you a basic introduction, then I am going to tell you various facts, then how you can use the GE in biopharmaceuticals, medical science, animal, vaccine production, DNA fingerprinting, also in insulin production, PCR, and you can also glow the plant in the dark environment. I have a very interesting topic along with these things. In vaccine area, I will tell you about the COVID vaccine. There are lots of scientists are actually making COVID vaccines by using the rDNA technology or genetic engineering technology. So, let me start with the first topic, genetic engineering. What is genetic engineering? My dear students, genetic engineering or genetic modification or genetic manipulation, sometimes we call it. It is a process of direct manipulation of organisms gene by using the recombinant DNA or rDNA technology to alter genetic makeup of the organisms. You can see here is a very interesting picture, though this is just a pictorial representation. Basically, we are trying to do this. This is your DNA, you are trying to add a new base. Okay. If it is A, if it is A, T, G, C, okay, you are trying to change something. Traditionally, humans have manipulated genomes by indirectly by controlling the breeding or selecting offspring or desired traits. See, when scientist Gregor Johann Mendel, when he has told his great heredity based understanding, see that time we are actually using this technology like breeding and other things. Now, in present day we actually successfully understood the molecular mechanism, the molecular basis. That is why we right now we actually able to do some changes in the molecular level. We actually using recombinant DNA technology, rDNA. That is what I have told you in my previous lectures. Now, if I tell you entire genetic engineering, this is not only about rDNA. Yes, genetic cloning, your gene editing, gene knockout, these things are also falling under genetic engineering. Cloning is much more relying on rDNA technology, but along all those things rDNA you can say is the bigger or must I can say rDNA technology is actually highly used. Okay. Now, gene knockout is a molecular biology technology where actually we do study the function of gene and removing the gene or observing the effect of gene on an organism. In editing is a process of altering the makeup by using CRISPR, for example, CRISPR or Talent or other technologies actually used in gene editing. Here in gene cloning, it is essentially the insertion of a specific piece of the desired DNA into a host cell, such a way that inserted DNA replicate and handed into a daughter cell during cell division. It is used for the creation of the copies or clones of particular gene. rDNA, all of you are aware what is rDNA. I will not again tell you, because in my previous whole lecture I have actually dedicated for rDNA, but still let me tell you if you have missed my previous lecture. So, it is a technology used of joining together DNA molecules from two different species. You have one species, you have another species. You are joining the DNA of two different species and you are trying to make a new combination, recombinant DNA. Okay. This will be recombinant, this will be inserted into a host organism to produce the genetic combination. Okay. This is something about our genetic engineering's entire landscape. Now, let me tell you few facts. You know, you have if you have listened my previous lecture, you have seen the first artificial that is a gene when it was actually developed. Now, let me tell you the first artificial or genetically modified animal was created in 1973, when Boyer and Stanley Cohen together to genetically engineer successful of the genetically engineered GE model. It is the first genetically engineered that is a animal rat. Then some of the most common animal tests are subject to the genetic engineering are mice, zebrafish, etc. Both are actually relatively short lifespan. So, any modification to their DNA can be assessed quickly for the growing of the animal. Right now, whatever the genetic engineering we are doing, we have to validate, yes, it is working. We are actually using either zebrafish or either we may use the such kind of mice. If I tell you more, this genetically modified researcher can build completely synthetic genome and they are able to conduct experiments on genome that do not actually exist in the nature. This is called a very big branch of called synthetic genomics. Many of the people are working. Genetic engineers can also today create entirely synthetic gene. I hope you remember human artificial chromosome. I have mentioned in my 10th lecture. Okay. So, HSC, it is the synthetic chromosome. For example, first synthetic chromosome was actually yeast was created few years ago. Now, in biopharmaceuticals, how we can use the genetic engineering in biopharmaceuticals. You see, my dear students, when you learn about this subject, I hope many of you are from pharmacy background or you might be from biotechnology background, because in our pharmacy in 6th semester only one subject is actually dedicated to biotechnology. I believe this subject should be taught in a great that is a way. 2nd semester, 6th semester, 4th semester, I think in this subject should be taught in much more that is classes, okay, because this field is actually growing very rapidly. All of you are aware about FDA. FDA is actually approving either small molecule or FDA is actually approving biologics. You know what is biologics? Small molecule, all of you are aware, basically drugs. Drugs means like molecules which molecular weight is almost we can say less than 10,000 Dalton. 10,000 Dalton. The molecules which molecular weight is less than 10,000 Dalton. Here biologics, these are the big molecule like vaccines, various proteins or various kinds of biological substances. Hai. So, these are the two areas, small molecule, yeah, here is our FDA. So, FDA is actually benchmark in whole about the world for approving the drug and other things. Now, you have to understand in our drug we call something as an generic medicine, or sometimes we also talk about the something called bioequivalence studies. Here we have to talk about the biosimilar. You know what, in this field every year you will be observing lots of new biologics FDA will approve. In around the world, huge number of biopharmaceutical companies, they are working relentlessly to develop new that is a protein or also new MAV, monoclonal antibodies as a therapeutics. They are doing very relentlessly. So, huge amounts of drugs that is biologics are actually approved. If you compare, obviously this will be much more higher, these drugs will be much more higher compared this, these are much more lower. Okay. But in future you will be observing huge amounts of biologics every year will be approved by FDA. If I tell you present day scenario, every year I can tell you that almost 12, 10, 8 number of biologics actually approved. Okay. For example, in 2020 I think 8 or 10, 12 we are I think given something. Okay. So, very few number I can tell you, I can summarize less than 15 every year, less than 15 number of biologics are actually approved by FDA. But in this case this is much more higher, this is more than 15, this is more than 15. Now, Why I am telling you all those things? Because over last 20 years various regulatory bodies like FDA, European organization and other authorities have approved approximately 95% of biopharmaceutical drugs. These approval are given for diseases like diabetes mellitus, various growth disorders, inflammatory condition and also neurological condition. With development in biotechnology, several drugs are now being produced for their biological process. One of the great example is the insulin hormone, which is really made in our pancreas as a gland located behind in your stomach. It allows your body to regulate glucose for energy. Glucose is a type of the sugar, all of you are aware. Now, due to advancement of your genetic engineering, it is possible to development of insulin to fight against diabetes. The bovine and porcine insulin actually that is derived from the pancreas of the animals, which is similar to human insulin. Okay. These animals, these animals or this pancreas from here were actually that is bringing, and then we are actually trying to get the insulin from cow and other, because their insulin is similar to us. This insulin are causing inflammation at the place of injection, which raised question of the safety. Thus, researchers produced recombinant insulin produced by the insertion of the insulin gene in the vector. This insulin safe and effective. Let me tell you in detail how they have done. See, you have some cow or human, both of them having insulin. So, in human, if insulin is getting there is a insulin deficiency, what we will do? In previous time, earlier time, cow, bovine and other other animals, their pancreas actually used to collect it, and from that pancreas the insulin actually collected, and that used to give to the human, so that their insulin level may increase. The problem is that it is not working correctly. It is not working correctly, the inflammation, other things are happening. Then the RDNA technology was actually introduced. So, in cow or in human, in there must be some gene, which will be responsible for making this insulin. There must be some gene. So, what they have done? They have taken out that gene under RDNA technology, that gene, and then plasmid or some vector. Let me write the correct word, vector. Okay. Then they have fused that gene with the vector like that, and then it was transferred into E. coli, and that E. coli actually produced the insulin, and that insulin can be given to the human. Okay. Right now, I will tell you more in my upcoming slide. Then also in 1972, Paul Berg made the first recombinant DNA molecule by combining the DNA molecule from monkey virus SV40 with the lambda virus. Since the discovery of gene therapy in 1980, it helped prevent lots of diseases and genetic disorders like Down syndrome, cancer, cystic fibrosis, Alzheimer's disease, Huntington disease, also various cardiovascular disease and viral infection, also such as in the AIDS. You can see how that is much right now we are actually depending on this technology called recombinant DNA technology, here recombinant DNA molecule. So, what they have done? They actually taken one virus, two viruses, they have fused the DNA from both the viruses, they have inserted somewhere, and they have got some new thing. In the upcoming year, gene therapy will play a very vital role in the treatment of many such diseases. We already have seen, and the advancement will made biotechnology and GE brought together in gene therapy into the limelight. Presently, many of you might be thinking that, sir, this technology is not so much popular. I will agree, because there are lots of new things is supposed to be discovered, but let me tell you, recently there is something called AlphaFold, okay, DeepMind of Google's one organization. They have developed one software called AlphaFold. It can predict 3D structure of protein in a very correct manner. Okay, AlphaFold. So, what we will observing in next few years, whatever I have told in my beginning of my lecture, that every year only 10 to 15 biologics are actually approved. Now, the great days are yet to come due to development of the AlphaFold. This is free software. You can predict the 3D structure of the protein, because ultimately you need the 3D structure of the protein, so that you can analyze that this protein will bind with which receptor. And you know, by using RNA technology, you will be able to make your desired protein, that is 3D structure of the protein. Okay. So, it is a very big field. Also, different approaches are actually used in process of the gene therapy. Firstly, normal gene can be inserted into the genome at a specific site to replace the defective gene. Secondly, the defective gene can be exchanged with the homologous recombination of the normal gene, and thirdly, there is a possibility of repair the defective gene through the reverse mutation, and lastly, expression of the gene can be altered. So, all the things are possible right now. In animals, in animals actually widely also used. In 1980, scientists are successful by add stably inherited new genes to the animal. Mice and fruit flies were actually one of the first animals in which testing was conducted. Later in 1982, Richard Plumilter and his colleagues discovered that the growth of the mice happened with the combination of the growth hormone of rat and this thing called metabolism gene of this mouse lead to the growth of the hormone production. This led to develop a new that is area in the field of hormone. Then one of the successful scientific breakthrough happened in the 1977, when scientists actually in Scotland they have cloned sheep using cloning technique. When I say sheep, I hope many of you might remember the story called Dolly, gene cloning was happened, and then CRISPR was actually introduced for the gene editing, whatever I have told. In 1919, first time a treatment was that is it was actually used treating in the patient of genetic disorder, basically CRISPR. If I tell you more about the genetic engineering in vaccine production, widely we are actually using widely. So, using genetic engineering techniques, vaccines can be developed by inserting genes from disease causing microbes into the harmless bacteria or viruses. See, this is a pathogen, this is a disease causing microbes, for example, like SARS-CoV-2. If you take some part of its genome, if you extract its genome and if you take some part of its genome and if you insert that genome into a harmless virus, what will happen? Harmless virus, this is here, and then the entire thing, if you inject some person body, what will happen? Obviously, this part when it will enter, it will translate, it will create some protein, which protein will be similar like this epitope of this virus epitope. So, what will happen? Ultimately, when the virus will enter to the body, your body will be fighting this, your immunity will be developed. I have few example. Right now, all of you are aware about the coronavirus SARS-CoV-2. This has created a very big challenge in front of us. For SARS-CoV-2, widely we are actually using. How we are actually using? For example, this is a virus. In that virus, there is something called spike protein, and its top part is called RBD. Okay. So, RBD systems actually widely available. So, what we actually try to do? We have to do some immersion and other things. So, before going to the, I am just trying to tell you how they are doing. So, clinical testing will be done. For example, this part you have taken this part, and you are trying to know that which part, this part actually RBD or spike protein, this part actually coming from which gene. You are trying to know, you have identified that gene, and then you have done some rDNA, and then you have done some clinical testing. You have taken that gene, then you have combined that thing with rDNA. This is a spike protein, this is harmless. From this virus, this spike protein or RBD region is the harmless part. So, you have taken that gene and you have applied the rDNA technology with that particular gene, and then whatever the formulation you have got in this form, emulsion other things, that formulation you have inserted in animal, in human for clinical testing. What will happen? First humoral human response will be happen. Okay. So, whatever the foreign substance actually entered, that thing will be first attacked by using your various antibodies. Okay. And also new antibodies will be created, and also you will be observing the cellular immune response, because ultimately your CD4 cell, that is killer T cell, helper T cell, these things will be come to the picture, and whatever the cells it has created some problem, that cell will be also destroyed. Okay, this is how it will work. Now, how? Here I have told you, you are actually using the RDNA technology. Now, how we are actually using? So, here you can see the patient, here you can see the patient. So, ultimately when the first dose will be given, these things will be called as an immunization period. So, during immunization period, you have seen in my previous this slide that these things cellular immune response. So, naive T cell, after entering of that antigen, this is antigen basically, after entering of this antigen, the antigen presenting cell, dendritic cell, it will engulf that thing and it will represent these things to the naive T cell. Naive T cell will inform to the CD8 cell, that is a killer cell, and also CD4 cell for B cell activation. Okay. So, you can see the B cell activation, B cells are actually producing antibodies, and those antibodies will be joining and binding with your these things spike protein, and then some of these antibodies will be saved as a memory cell. After that, in future, the next stage, when the actual virus may enter in your body, your entire system will be activated. Ultimately, effector CD4 T cell, CD8 cell, that is killer cell, will be come together and it will destroy that cell which cell was actually infected by the virus. And the other side, your antibody, whatever antibody is actually produced, that antibody will be do opsonization or neutralization, it will be done. You can see the virus actually neutralized by antibodies. This is how our in COVID period also genetic engineering playing a very big role. In fingerprinting, we are also using. So, in DNA fingerprinting, how DNA actually first extracted from the cell, then extracted DNA actually cut into smaller pieces with special enzyme, then the DNA pieces are placed on a gel that uses electric current to push them through the gel and separate according to their size. The larger DNA pieces are remain closer to the top, while smaller one move to the bottom. Yeah, and then the separated DNA pieces on the gel are actually stained and reveal their unique band with the DNA fingerprint. See, this is actually called electrophoresis system. I have told you in my PCR lecture that how we can use electrophoresis. So, now by using this picture, if you compare with this picture with the known suspects, then you will be able to identify whether somebody is responsible behind a crime or not. Here is a very important, very beautiful picture. You can see this picture of 1950 and this picture is now. To make 0.05 kg, of insulin, you have to take this much 4535 kilogram of pig pancreas. Can you imagine? You have to sacrifice a lots of pigs, so that these are the pig's pancreas, so that you can get such small amount of insulin. But today bacteria is actually producing insulin without harming anybody. Okay. Now, another example is here is the picture how it is actually happening. Here is the pancreas, we have DNA strand, we are actually taking that gene which is actually responsible for insulin creation, and here is the bacterial plasmid. We actually put that insulin gene into that is a bacterial plasmid, and then we actually add that thing into the bacteria, and then when it will express, it will make the insulin. We also using the same concept in PCR, polymerase chain reaction. I will not discuss too much about for today due to the shortage of time, but let me tell you in this I have a separate lecture on PCR in my upcoming days, you will be observing, do not worry. So, here you actually make multiple copies from 2 to 4, 4 to 8, 8 to 16. If a certain substance is present, then it will be give you such kind of graph. If a certain DNA, for example, certain DNA is not present, it will give you such kind of graph. So, based on the difference between graph, we can tell. I will tell you more in my next lecture. You can take also screenshot of this. I have a separate lecture on PCR and RT-PCR. Do not worry. And last, by using genetic engineering, you can glow the dark plants. You can see these two scientists, their dream become true. This news was published in New York Times in very recent in 2013. So, these are the things whatever we have covered. We have learned about genetic engineering, various facts in biopharmaceutical area, medical science, animals, vaccine production, DNA fingerprinting, insulin production, PCR, how we can use our genetic engineering. In next lecture, I will tell you about interferon therapy, how you can use rDNA in interferon production, clinical toxicity and various marketed formulation. So, that is it from my side on today. Thank you all of you for joining with me and listening this lecture with very much patience. So, we may meet in our next lecture. God bless you, keep learning, keep shining. Thank you all of you for joining. We may meet on next lecture. All the best.